Alicia.
So who knows what's going to happen in 10 years? So I'm not going to go that much into the future. But as I have been working more with the crop community, where they have not only reference genomes, but pan-reference genomes and all these beautiful genomic resources, I can tell you that their GBS and RAD are still useful to generate data sets at the population level, or to look at QTL variation, and to perform GWAS and this kind of analysis that really need a lot of samples. And they work very well in combination with reference genomes in that sense. So I think there will be still room for GBS for that type of questions that are population-based or related to GWAS or PTLs, this kind of questions that use those tools in conjugation with reference genomes. So, and this is just looking at what is doing the crop community, thinking that, well, they were doing genomes and other stuff before the wildlife community, so to say. So I suppose that the natural step will be to do something similar to what they are doing. How many years would that last? I wouldn't know. Thanks.
I guess I have a quick follow-up. Do you think part of that is because of the size of plant genomes? Like, is the cost of re-sequencing 100 plant genomes just a lot higher?
Not necessarily, because not all plants have huge genomes, although some crops do indeed. But it's more about the number of samples and the questions you want to ask. So imagine you are doing plant breeding, or you are scanning for loci under selection related to, I don't know, drought resistance or whatever. Then you want thousands of individuals to be analyzed of different populations of sources, or as part of an experiment. And it's really what they are, or we are doing. It's just not a couple of genomes. It's good genomes and then hundreds of samples of different individuals.
I totally agree with Alicia. Yeah, who knows what will happen in the future? Yes, I agree. But I think in 10 years, I agree with Alicia. It's really about what is your question, right? And what is the data set, and you need to answer the question. And I think that more and more these days, there's kind of like deep sequencing at the whole genome scale, but there's also, a kind of like emerging, set of questions that people are going to become more interested in, is like spatial questions, right? And so for spatial sampling, if you have a nice reference genome, it will allow you to maximize the kind of sampling, maximize your sequencing effort by obtaining a broad spatial sampling, which, if you have broad spatial sampling with whole genome sequencing, and you're really just interested in kind of population or demographic processes, then you're just wasting a bunch of effort, right? So I don't think that will change necessarily, like in the near to kind of near distant future.
Melanie, do you? Oh, go ahead. Oh, go ahead, Jon.
Melanie, you go.
I can just quickly say, yeah, I agree with what's been said so far. And I'm just thinking about, I did a big landscape genomics project recently where landscape genetics using genomic data, where basically you generate all this giant data sets, thousands of SNPs, and then you condense it down to a single genetic distance metric between individuals. And that's what you're using in your analyses. And so I think that if you're using genomic data for population genetics, where you are really condensing down to these single metrics, that a lot of times GPS data is more than sufficient to do that kind of analyses. I think when you want to start thinking about whole genome sequencing is when you're, looking for outlier loci, you're looking at adaptation, you're, wanting to look at allele frequency spectrums, and you're starting to use the data for more than just how genetically similar are two individuals from each other.
Yeah, Jon, go ahead.
Yeah, I'd echo basically what everyone said. I think, even with, we end up with perfect genomes for every species. For a lot of species, once you're kind of above probably 100 megabases, like you have significant cost savings by doing, reduced representation approaches. And so I don't think RAD-seq is going away anytime really soon. I will say that, or I'll at least take one chance to plug a different type of sequencing that my lab works on, which is called express exome capture sequencing, which if you are looking for selection in wild populations, like maybe a good way to go. And feel free to reach out to me if you're interested in more about that. But I do think that RAD sequencing is going to be here for a while. I mean, people still use microsatellites for stuff, and RAD sequencing is way better than that. So, and it's almost as cheap now. So I think it'll be here for a long time. It has lots of uses. And so don't be afraid to put in grants to do RAD-seq projects. That, go for it.
Awesome. Well, thank you so much to our panelists. We're out of time. We'll be posting this presentation on the website. I'll also be adding the links that Isaac sent. Those are really helpful. We'll put them on there as well. Yeah, Alicia.
Can I just please say something that there was no opportunity to say before? And I think this is important for fish, and I suppose there's fish in the oceans. There are fish. Don't just filter paralogs. We do it because we are interested in population genetics with orthologs, but I think that paralogs have a lot of cool information, and we already have them, and we are just putting them into the bin trash. And there is information there, a very cool one. And I think this will be especially important for fishes because I think their genomes are weird as lots of plants. So I couldn't leave without saying that. I want to shut up. Thank you.
No, that's a great point. I think there's a lot of like copy number variation work that's starting to come out that, yeah, we'll definitely have to look at more in the future. Great. Well, yeah, thank you all so much. Fill out the survey if you haven't yet, if you attended, just so we know who's here. And we'll see you hopefully in 2 weeks for our next panel seminar on whole genome sequencing and the considerations for low and high coverage genotyping. All right, thank you all so much.
Thank you.
Thanks.
Bye, thank you guys, yeah.
Embrace missing data.
Embrace missing data, we'll make a shirt for sure.
I want a t-shirt.
Yeah.
See you,
I'll work on
everyone.
it.
Yeah.
All right, great to meet you. Good to see you again, Isaac.
Yeah, you too. You too.
Bye, Isaac. Bye, Jon. Bye,
Bye,
Daniela.
Alicia. Bye, Jon.
Hey.